Hi everyone! Today I'm going to be explaining the 4 levels of protein structure and the different bonds involved in each level. Starting with the primary structure, this is basically just the sequence of amino acids in a protein chain. I like to think of it kind of like letters in a word because changing even one amino acid can completely change the protein later on. The amino acids are connected by peptide bonds, which are covalent bonds, so they're strong and help hold the chain together. At this point, the protein hasn't folded yet, and it's just a straight chain. Next is the secondary structure. This is when parts of the chain start folding into patterns like alpha helix or beta pleated sheets. These structures form because hydrogen bonds develop between atoms in the backbone of the protein. So instead of the side chains interacting, it's mainly the backbone stabilizing itself through hydrogen bonding. This creates local folding patterns, but not the full shape of the protein yet. Then we have the tertiary structure. This is the full three-dimensional shape of one polypeptide chain. This is where the side chains or the R groups start interacting with each other. Nonpolar groups usually move inwardly away from the water, while polar and charged groups stay more towards the outside. Different interactions help stabilize this structure, including hydrogen bonds, ionic bonds, hydrophobic interactions, van der Waals forces, and disulfide bridges. This final shape is really important because it determines the protein's function. Finally, we have the quaternary structure happens when multiple polypeptide chains come together to form one functional protein. Not all proteins have this level, but many important ones do, like the hemoglobin. The same interactions from tertiary structure help hold these subunits together, except now the interactions happen between separate polypeptide chains instead of within just one chain. And overall, each level of protein structure builds on the previous one, allowing proteins to fold correctly and perform their specific functions in the cell. Thank you so much.